Hey, Bio30s, and today we're going to be starting a brand new unit on molecular genetics. Today I'm going to be talking and walking you through the history of the discoveries about DNA and how we kind of came to learn that DNA was in fact the source of hereditable material in organisms. Because for the longest time, a lot of people believed that it had to do with proteins rather than nucleic acids. So we'll get started on that today before we actually start discussing bigger topics of molecular genetics, including DNA replication and protein synthesis. So for today, a little bit of a history lesson to get you started. First of all, molecular genetics in your textbook is going to be on chapter 18, pages 622 to 665 in the textbook. So just so you know, that is the chapter you're going to be looking at in your textbook. So let's start discussing a little bit about the history about how we discovered what DNA actually looks like. How do we know that DNA looks like this spiral staircase, this famous double helix model that you may have heard of about DNA. So first of all, DNA, if you want to take a quick peek, here's a little video that's going to break down for you DNA and what's it all about, just so you kind of have a little bit of a heads up before we move into this conversation. You should be kind of familiar with it, but if you wanted to give this quick little video a watch, it'll give you, I guess, a better idea about DNA in general. But DNA, generally speaking, again, it stands for what we call deoxyribonucleic acid. It's one of two forms of nucleic acids that we find in cells. The other type of nucleic acid is known as RNA, which is ribonucleic acid. And the names are different because of the sugars that are found in these molecules, as I will get to in a few minutes. Now, nucleic acids, these acids that are basically one of the four major macromolecule groups that we discussed all the way back in Bio 20, along with carbohydrates, lipids, and proteins. Nucleic acids were first discovered back in 1869 by Frederick Michner, who is a Swiss physician. He discovered them under a microscope. He stained the microscope and realized that there were specific substances in the nucleus that he could not identify as being any other type of substance like a protein or anything else. So he was the one who coined the term nucleic acid, but he didn't really know much about them. And just so you know that when he discovered nucleic acids, we're going to be in about 2 units from now, we'll be discussing classical genetics and a gentleman by the name of Gregor Mendel, who discovered how genes pass on their traits from generation to generation. This discovery actually happened after Mendel had discovered how the mechanism of inheritance. He didn't know what was causing those mechanisms of inheritance, but Michner was the first guy to discover that there was a substance called nucleic acid, which we now know is the source of this heredity. At the time, though, he had no idea what these nucleic acids were doing. Step forward about 31 years to a guy by the name of Phoebus Levine in 1900. He discovers that there are, in fact, two types of nucleic acids, which I just mentioned to you a few minutes ago or a few seconds ago, DNA and RNA. And he also discovered that they were polymers, just like proteins, like carbohydrates, like lipids. They're long chain molecules that are comprised of repeating subunits known as monomers. And he discovered the monomers were known as, well, he called them nucleotides. That was sort of the name that he coined for them. And a nucleotide is comprised of three things, as he discovered. He discovered that there was a 5-carbon sugar there, which is why you have DNA versus RNA. Deoxyribose is the sugar in DNA. Ribose is the sugar in RNA. That's why you've got a D versus an R there. There's also a phosphate group that's part of that monomer group. And there's also a nitrogenous base. Now, the nitrogenous bases, these would be almost analogous. They're not quite proteins, but again, proteins have nitrogen in them. These are bases that contain, obviously, nitrogen in them. But there were two groups that he discovered. There were one group called purines, and the purines were basically double-ringed nucleic acids, double-ringed nitrogenous bases. And they were comprised of two different ones. One's called guanine and one's called adenine. He also discovered there were other ones called pyrimidines, which are single-ringed structured nitrogenous bases. That would be cytosine, thymine, and uracil. Now, uracil is an oddball, which I'll get to a little bit later. It's only found in RNA. thymine is only found in DNA. The other three, guanine, adenine, and cytosine, are both found in DNA as well as RNA. So he discovered that in 1900. And again, I'm not going to discuss him in great detail, but there is a slide down here. Thomas Hunt Morgan, I'll be discussing him more when we get to classical genetics, but he basically began the first experimentation on genes, on DNA, to basically figure out, where genes were located on chromosomes. And he kind of did these experiments, and scientists did not know at that time whether it was DNA or proteins that were involved in the genetic transmission of traits from generation to generation. So it was basically how this was discovered were through about three different groups of scientists, which I'll be getting to right away right now. The first one was in 1928, Frederick Griffith. He wasn't really working with DNA. He was working on actually experimentations because he was trying to figure out a way to kind of prevent pneumonia outbreaks or epidemics in London. And when he was doing an experiment, and there was a great little diagram here on the slide, that he discovered something called the transforming principle. That's what he called it. And what he discovered was he made a mistake. He actually, if he gave a certain bacteria to mice or to rats or to mice, like it was mice, he could give a pathogenic strain of a bacteria that caused pneumonia to a mouse and it would die. Pathogenic just means disease causing, right? He would then give non-pathogenic strains of bacteria to the mice. The mice would not die because they weren't ones that caused pneumonia. Then he would heat kill pathogenic bacteria. He would break them apart and boil them and break them apart. When he did that, the mice also would not die because those bacteria were presumably dead. But then he made a mistake. He put some of these heat-treated pathogenic bacteria in a mix with bacteria that didn't cause pneumonia. And what he discovered was, even though he put those dead pathogenic bacteria in the mix by accident, what he discovered were the non-pathogenic bacteria began causing the disease. So he didn't know, he just knew there was a transformation going on with these non-pathogenic bacteria, but he didn't know how or why. Now, we obviously know that it's the genetic information of the dead pathogenic bacteria were somehow being incorporated into the live bacteria, the non-pathogenic ones. We discussed this in the last unit in cell division when we discussed the use of a conjugation, use of pilus or pili between bacteria to swap genetic information to cause changes in their DNA. So he discovered this, but didn't really know what was involved. So he wanted to kind of conduct more experiments. Unfortunately, World War II broke out, and he went to war and died. And he never actually finished his experiment. So this transforming principle idea that he discovered in 1928 sort of remained dormant until after World War II. Then come along a group of scientists by the name of Avery, MacLeod, and Macklin, and they wanted to prove once and for all what was causing this transforming principle. They believed it was, in fact, DNA that was doing it and not proteins, like a lot of people at that time believed, because a lot of people still believed in the 1950s that it was proteins that were involved in the transmission of genetic information from one generation to the next. So what they did is they conducted an experiment. They basically heat-killed some pathogenic bacteria. They treated heat-killed bacteria that were pathogenic with two different groups of enzymes. They took one group of this killed pathogenic bacteria, and they basically gave them what were called proteases, enzymes that would break down proteins. Like we talked about this in Bio 20, enzymes like trypsin and chymotrypsin and pepsin and erypsin. They basically broke down the proteins in these bacteria. When they did that, and they put these bacteria that they had treated with these proteases into a batch of live non-pathogenic bacteria, they still found that the transforming principle still occurred. So they felt that it wasn't the proteins that were doing it. Now, when they used enzymes that broke down nucleic acids, like nucleases, and they tried the same experiment with these dead pathogenic bacteria, they discovered that, lo and behold, the transforming principle didn't occur anymore. So they felt that after that experiment that they had clearly defined that it was DNA or nucleic acid that was actually transforming these live bacteria into ones that could cause disease. But despite that great experiments, there were still some doubters. There were still some people who believed that somehow the proteins were involved. So another group of scientists came out about 8 years later, because Macklin and his boys and Avery and MacLeod did their experiment in about 1944. 1952, two rock stars in the history of genetic research, Hershey and Chase. They came out with a different experiment to once and for all prove that it was DNA that was causing the transforming principle. So instead of using enzymes, they used radioactive isotopes. And you can see here, there's a sort of little diagram right down there. They use what were called bacteriophages, which are viruses that infect bacteria. And what they did is they radioactively, targeted or labeled parts of these bacteriophages. So what they did is they basically took the protein coat, the outer layer of the virus that basically is the outside, analogous to the cell membrane, because viruses don't have a cell membrane. They marked that protein coat using radioactive sulfur. Then what they did is they took the DNA that was inside the virus and they tagged that or labeled that with radioactive phosphorus. So they had two radioactive isotopes, and what they wanted to see is which one of these, if either, was going to be injected into the bacteria that the virus was invading. And lo and behold, after they did those labels, what they discovered was, guess what? The DNA that was labeled with the radioactive phosphorus, it was found inside these bacteria. None of the radioactive sulfur that represented the proteins of the virus were making their way into the bacteria when the viruses invaded the bacteria. So this finally, without a doubt, concluded that it was basically DNA that was actually used to reproduce or to transform one bacteria into another, in this case to reproduce viruses inside of a bacteria, in a cell of a bacterium cell. So that was an extremely important experiment that was conducted that finally, so by 1952, finally science has got it figured out that it's actually DNA that's actually causing that. Now, what about some of the research that was going on with the structure of DNA? Now, back to 1900, we talked about Phoebus Levine, and he discovered that there were nucleotides. He realized that there were sugars, there were phosphate groups, there were nitrogenous bases. So what was the configuration? What did this actual molecule look like? Well, some of the early research on this was conducted by a guy by the name of Gerwin Chargaff, who was a Ukrainian-American scientist in the late 1940s in America. He was basically conducting experiments. And what he discovered through his research was, after he began taking sections of DNA, he found that there was a sort of a pattern that was being followed. He realized after, tons of research and tons of examples of DNA, he always found a pattern in the DNA. And that pattern is in this little diagram down here, and it's known as Chargaffs Rule. And this is a really important rule in genetic science, or in molecular genetics, is that he discovered that the nitrogenous bases adenine and thymine were always found in equal quantities in any piece of DNA that he looked at. He also discovered that if you knew how much cytosine was there, you also discovered that the amount of guanine was equal to the amount of cytosine. So what he discovered was that A equals T and G equals C. That's what he discovered. So there was a pattern between the adenine and the thymine and the cytosine and the guanine. Now remember, I did mention uracil earlier. Uracil does not play a role in DNA. It's only discovered in RNA. So for now, we're just going to ignore that uracil. We'll get more to uracil when we discuss protein synthesis later in the unit. So, for example, and here's how I remember this. You think of one of the great rock bands of the 20th century, AC/DC. So what you do is you write down ATGC right here at the bottom of my diagram, ATGC, or my notes. That is going to be an indicator that A equals T, G equals C. So how does that play out? Well, I'm going to give you an example of this. So I'm going to erase part of my board here as I knock my board off the table. Holy smokes. I knew that was going to happen. Live television. I love this live aspect. You just never know what's going to happen. I got to hold this so it doesn't fall over. See, it's going to fall over again. This is the stuff you don't normally see on my videos. This is why I never touch my board, because it is very precariously balanced on the table. I just have a sneaking suspicion this is all going to fall down. Okay. Whew. We survived that. That was the toughest part of the lesson for me right there. Okay. So here's what I mean. If I know, essentially using Chargaffs Rule, if I know any percentage of any of the nitrogenous bases in DNA, I can calculate the percentages of all of them. So for example, let's say I've got 14% of my DNA. I've got a section of DNA that has 14% adenine. So what I'd like you to try to do based on Chargaff's rule, and remember A equals T, this is Chargaff's rule, A equals T, G equals C, I want you to calculate the percentages of all the other nitrogenous bases. So how would I do this? Well, I'm going to get you to pause the video right now, see if you can figure this out before I spill the beans on the board, and I'll give you the kind of the rundown on how to do this. Okay, so here's how we're going to do it. We know that A equals T, so I know that T has to be 14%. So that means that right now I've got 28% of my DNA is basically comprised of A and T, which means that if I have 100%, if I take 28% away from 100, that's going to leave me down here. The remaining 72% of my DNA should have either a G or a C nucleotide or nitrogenous base in it. So if that means that there's 72% for these two, I know they're equal. So I'm going to take 72 and I'm going to divide it by 2, which means that I'm going to get percentages here for the G and the C of 36% each. Now there's another way you can do this. This is a kind of a variation that I like to use. I also like to do it this way. I know that A plus G or A plus C or T plus C or T plus G, all of those numbers are going to equal 50%. So to do this even quicker, using that analogy, I'm going to just erase the board. I'm going to do it another way. I'm going to do the exact same. I'm going to do another example. So I'm going to give you this case. I'm going to give you G. G has a percentage of 22. So how can I calculate this very quickly? Well, I know that C is going to equal G, so that's 22%. Now let's use this variation instead. I know that G plus either T or A is going to equal 50%. So I know that 50 minus 22 is going to give me 28. That's going to give me 28 and 28. And again, how do I know I've done this right? Take all 4 of your numbers and they better equal up to 100%. If they don't, you're not following Chargaff's rule. Here's a little bit about what he did, some of the original, some of his original data is up on that slide there from one of his original papers, just to kind of show you sort of how he came about it. You can see sort of the data that he had. It was never 100% accurate, but he was always within scientific error on his thing. So what does this mean? Well, in the grand scheme of things, this all connects together when we finally get to the final model of DNA, which was devised by, usually it's accredited to 2 guys, Watson and Crick, but Rosalind Franklin was heavily involved as well. Rosalind Franklin kind of got screwed over on this. Quick little video if you want to watch about Rosalind Franklin, because she really is sort of the rock star behind the theory of the model of DNA, the double helix model. It was her work that sort of led the way for Watson and Crick to make their discovery. Yet everyone thinks about Watson and Crick. They don't normally think about Franklin when they think of the discovery. I think that's changing now a little bit now as people are beginning to realize what her contribution was. But she basically took this famous photograph right down here, an X-ray photograph, and she basically, that photograph got into the hands of Watson and Crick without her knowledge, and they were able to use that photograph to basically really quickly run through and make a draft of the model of DNA. And they basically came up with the double helix model, which basically got them a Nobel Peace Prize, in science. And unfortunately, Franklin didn't get it for a couple of reasons. One, because she wasn't really considered to be the person who came up with the photograph, I guess, or the idea, even though she was. And she died. She died very young, early on, because of all the, potentially because of all the X-ray photography she was doing. She died of cancer at a very young age. I think she was only like 38 years of age when she died in 1958, before the the Nobel Peace Prize was awarded to Watson and Crick. But her work, basically, she noted a couple things about the structure of DNA. She realized that the DNA had a helical, like a spiral pattern to it, with two regularly repeating patterns in it. She also discovered that the nitrogenous bases were inside the molecule, and the sugar and the phosphates were on the outside. So she learned all of that from her photograph, and then that photograph was sort of, they ran with it. Watson and Crick ran with it in 1953. Came up with their double helix model, and it was announced and published in a paper in 1963. Franklin published it in the same journal, but was sort of right after them in the journal and kind of got a little bit kind of bowled over with the discovery because Watson and Crick published apparently earlier in the book, and it just looked like her paper was kind of supporting what Watson and Crick had said. So, and she was a woman. She got kind of bowled over because she was a woman, in the 1950s in science, and who would listen to a woman in the 1950s, which is horrible, but that was unfortunately the political reality in the 1950s. So anyways, they discovered that the DNA molecule, and you can see our little double helix model right over there with my little thing right there. It's like a spiral staircase, and the inside of the stairs, the actual rungs of the ladder are the nitrogenous bases in the middle, and then the handles of the ladder are going to be your sugars, which are in red, and the phosphate groups, which are in blue. So in this diagram up on the board, my nitrogenous bases are green, my sugars, my deoxyribose is in red, and my phosphate groups are in blue. All of these that were originally, that were discovered as part of the nucleotides back in 1900 by Phoebus Levine, finally in 1953 they figure out how they're all arranged together. Now you can see that I've got some nitrogenous bases here. This kicks into Chargaffs Rule. The reason why the percentages of A was always equal to T and G was always equal to C is that you had these bases, these nitrogenous bases that would always pair up with each other. So because of Chargaffs Rule, and that felt isn't working, so I'm going to throw that one away. This felt, so here, I've got these little, and by the way, they're held together in the middle by little hydrogen bonds. That's what that double line is. But G is always going to combine with C, so C is going to be over here. T always combines with A. C always combines with G. And A combines with T. So you'll notice that on a strand of DNA, the code on one side of the strand is going to be complementary and also different than the genetic code or the nitrogenous base on the other side. And it's actually these letters, these A, T's, G's, and C's that are actually the genetic code that your DNA reads to actually, give you blue eyes or to give you hitchhiker's thumb or non-hitchhiker's thumb, or allows you to code for a protein, as we'll get to later in the unit. So again, A to T and G to C represents what are called complementary base pairs. And one other thing you'll notice about the diagram is I've got two strands to a DNA molecule. I've got this, this strand here and this strand here, which are separated by this hydrogen bond. The strands are in opposite directions from each other. The configuration is in an opposite configuration. This way, the phosphates are pointing in one direction, and this way, the phosphates are pointing in another direction. Now, those phosphate groups represent a specific end of the DNA. The side that has the phosphate group pointing in that direction, that is called the 5' end of a DNA strand. The opposite end that's pointing away from the phosphate group is called the 3' end. What you'll notice in this diagram, and I've drawn this incorrectly, I drew it incorrectly. My goodness, I'm glad I'm catching my mistakes today. Technically, this is my 5' end here. So you'll notice that the 5' end here is opposite the 3' end on the complementary strand. And for that reason, the DNA strands are considered to be anti-parallel. Now that's going to be a very important concept when we discuss our next topic, which is going to be DNA replication, which I'll get to in our next video. But for now, just be aware that DNA, when it's laid out, the 5' and the 3' end here means that I've got a 3 and a 5' there. I'm never going to see two 5' ends opposite on the same side of a DNA strand. It's going to be 5 to 5, 3 to 3, like so. That's anti-parallel. All right? Quick little video there, a little cartoon for you there. about DNA. A couple of last concepts about DNA. Just a couple more things to kind of go through before we finish up our conversation today, and then we're done. I guess I already pulled it off. I pulled it off the board too early. Hey, it's on the bottom of this slide. Hey, well, we'll get there. I'm full of surprises today. There we go. Mitochondrial DNA. Let's talk mitochondrial DNA. First of all, you've been up till now thought that DNA is only found in a nucleus. Wrong. There are organelles that have DNA as well. Chloroplasts in plants have DNA. Mitochondria and all organisms that have mitochondria has DNA. Now the question is, how did it get there? Give me 2 seconds and I'll explain that. But what is mitochondrial DNA and why is it important? Mitochondrial DNA really doesn't have anything to do with coding of proteins in the nucleus or heredity or anything like that, or passing on genetic information from generation to generation. But it's found in humans. It's found in mitochondria, and that mitochondrial DNA is important because it's only passed down by your mother's side of the family. So women can only pass down mitochondrial DNA. Why is that? Because the egg has mitochondria in it before a fertilization event occurs. In a male sperm, which we'll get to in the next unit, in the reproductive unit, male sperm, all the mitochondria are outside of the head of the sperm. They're in a little section called the midpiece, which is right below the head of the sperm. The genetic information of the sperm is in the head. So when that sperm enters the egg to actually fertilize it, none of the mitochondrial DNA get into the egg from the male side. So only women are passing down mitochondrial DNA. It's always the maternal bloodlines that are passing it down. Now, why is that important? It's a lot easier for us to track long lineages if you're only dealing with the maternal side of the family. Always the mother passing it down from generation to generation to generation. And for that reason, scientists will use mitochondrial DNA to actually get better assessments as to old lineages. We're talking like old, like prehistoric, thousands, if not tens of thousands of years old, even as maybe as much as 100,000 years. We can see lineages and where humans have come from a lot easier using mitochondrial DNA than using nuclear DNA. So that's why mitochondrial DNA is quite fascinating. And the other thing about mitochondrial DNA is that it's basically as I'm getting the phone call live on the air, we're gonna kill that. Mitochondrial DNA. The reason why it's there is scientists believe that at one point mitochondria were actually a separate one-celled organism that actually entered another cell and entered into something called endosymbiosis. So you literally had one cell engulf another cell, and it didn't kill that cell. It actually became they entered into a symbiotic relationship with each other. So that's sort of the running theory, that mitochondria at one point were separate organisms, that they actually got incorporated into what would become eukaryotic cells and actually became part of what every cell has. So over 1 billion years ago, when eukaryotic cells first formed, it's believed that in a lot of cases these organelles might have been other organisms. In some cases, they might have been other organisms like chloroplasts, like mitochondria. So that's kind of a cool theory behind why there's DNA in mitochondria and chloroplasts. Before I finish up, 2 more topics and we're done. RNA, we've talked a little bit about RNA today. That's the ribonucleic acid. How does it differ from DNA? In a couple of major ways. DNA is double-stranded. RNA is single-stranded, so it's only got one strand. The sugar in RNA is ribose as opposed to deoxyribose. And RNA does not have the nucleotide thymine. Instead, it has the nucleotide uracil. So instead of having ATGC in RNA, you would have the abbreviation AUGC. A's will combine with U's and G's will combine with C's, as we'll discover when we get to protein synthesis later in the unit. There's also 3 types of RNA. There's messenger RNA, mRNA, transfer RNA or tRNA, and ribosomal RNA or rRNA, all of which I will discuss when we get to protein synthesis later in the unit. Final topic, genes versus genomes. You hopefully now know what a gene is. A gene is a small section of DNA on a piece of chromatin or a chromosome that's going to code for one specific trait, or as you'll find out later in this unit, they also code for single proteins as well. That's what a gene is. A genome is essentially all the genetic information of an organism that's found in every single one of its cells. So it essentially, when we talked about karyotypes earlier this year in the cell division unit, and we remember what a karyotype was, it was a microscopic photograph of all 46 of the human chromosomes. That karyotype is essentially the genome. That's all the genetic information of that organism that's found in every single somatic cell, in this case of a human, if there's 46 chromosomes arranged into 23 pairs. So that's what a genome is. Now, a genome, we discovered what the genome was in humans from 1990 to 2003 with the Human Genome Project, where they basically wrote down the code of every single nitrogenous base, I don't even know how many. There were 30,000 genes they coded and 3 billion base pairs. So 3 billion of these nitrogenous base pairs were transcribed. This was a huge project involving 20 universities, 7 different countries, and the process did speed up, but it still took 13 years. Even as computer processing was getting stronger and faster, it still took them 13 years to code for every single one of the 3 billion base pairs that makes up the genome of a human organism. This was done through a process called DNA sequencing, which allows you to identify specific nucleotide sequences in sections of DNA. And and just so you know that there is a technique out there called the Sanger method. You would need to know that. It was developed in 1977, but essentially now with computers, we can basically read about 400 million base pairs in roughly 10 hours. So the technology now has gotten a lot quicker to actually do a DNA sequencing on large sections of DNA in a very short amount of time. Just how big is the human genome to compare it? Okay, so right here you got a diagram if you got your notes in front of you here. To the right is the complete genome of a bacteriophage known as PIX174. It's a bacterial virus. It has 9 genes, which code for 9 proteins in it. The longest gene is highlighted in yellow. So all this writing in this little box are all the base pairs that make up that genome of that virus. The human genome would be 1 million of these boxes. There'd be 1 million of those boxes full of the same number of base pairs to basically show all of the genome of a human, every single base pair that makes up the human genome. That will comprise all the genetic information found in all 46 chromosomes in a human karyotype. And that's basically it. I've got you a preview of our next topic, which is going to be DNA replication. But for now, I want you to go over to the website, go and take a look at the description below this video, and I want you to complete the molecular genetics DNA versus RNA structure question booklet and get that to me for next day. That's it for now. We will be back next time for more discussions about molecular genetics when I discuss the fascinating method that DNA can replicate itself. See you soon.